Let's go over a couple more examples. Okay, so we're going to take a look now at glucose. So we're basically starting with the same concentration that we had in our other example. We have 4 glucose molecules in 100 milliliters. All of that's going to filter in. When we go down here, what is going to happen is that all of that glucose is going to get reabsorbed into the bloodstream. As a result of that, we are not going to excrete any glucose. Okay, so looking at this example, let's see if you can figure out what the glucose clearance is. So take a moment to think about that. Pause if you need to. Okay, so hopefully you figured out that the glucose clearance is zero milliliters per minute. We started with our 100 milliliters that had 4 glucose molecules in it. We ended up with 100 milliliters that has 4 glucose molecules in it. So we did not get rid of any glucose. We did not clear any plasma of glucose. Therefore, our glucose clearance is 0 milliliters per minute. I have a couple more examples to go through. This one, we're going to look at this molecule called urea. Urea is going to start at that same concentration, 4 in this 100 milliliter box of plasma. That's going to filter through, and then what's going to happen is we are going to reabsorb half of that urea that had filtered in. Okay, so we reabsorb half and we end up excreting half of it. So in this example, what do you think the urea clearance is? Pause if you need to, think about it. Okay, so the urea clearance is 50 milliliters per minute. Okay, so just kind of thinking through this, basically we are excreting 2 urea per minute that was given, okay? That 2 urea is going to correspond to 50 milliliters of plasma. So it's as though you took 50 milliliters of plasma and you cleared it completely of urea. So that's why we say that the urea clearance is 50 milliliters per minute. Last example here, penicillin, okay? So penicillin, again, we're gonna start with that same concentration, 4 in this box of 100 milliliters of plasma. That plasma is going to filter into the tubule. And then what happens is that we actually end up secreting additional penicillin into the filtrate here, okay? So at the end, we end up not reabsorbing any penicillin, okay? And we end up excreting actually more penicillin than was originally filtered. So in this example, although it's not shown right here, what we're doing is we are excreting 6 penicillins per minute. Based on that number, let's see if you can figure out what the clearance is of penicillin in this example. So take a moment, hit pause if you need to. Okay, penicillin clearance is 150 milliliters per minute. So again, explaining this, remember that we had 6 penicillins excreted per minute. That 6 penicillins corresponds to a volume of 150 milliliters of plasma based on our concentration of 4 penicillins per 100 milliliters, okay? So it's as though we took 150 milliliters of plasma and we cleared it completely of penicillin in 1 minute. So the penicillin clearance is 150 milliliters per minute.